Good afternoon. I'm Dr. Scott Stromatt, the Chief Medical Officer at Genetx. I'm joined today by Dr. Barry Kravis. She is a professor of pediatrics, neurological sciences, and biochemistry, and is a co-director of the Angelman Syndrome Clinic at Rush University in Chicago. We're going to present the preliminary data from the phase 1 trial of the first 5 children treated with GTX 102. Just in disclosure, GTX 102 is an investigational drug and is not approved by the FDA nor any regulatory authority. The data presented here are interim data and do not provide definitive conclusions on efficacy or safety. Substantially more work is required to prove whether or not GTX 102 is safe and effective. GTX 102 is an antisense oligonucleotide, or an ASO. ASOs are generally single-stranded synthetic molecules composed of DNA and RNA. They target the RNA, and that alters RNA expression, and therefore protein production. In Angelman syndrome, there is a missing or dysfunctional UB3A maternal gene. GTX 102 activates the paternal copy that is typically silenced. In neurotypical individuals, you can see here in the top right, the maternal gene is on and produces UBE3A protein. The paternal gene is turned off by an antisense transcript. In individuals with Angelman syndrome, the maternal gene is dysfunctional and the paternal gene is silenced. That silenced is also called imprinting. The goal of treatment with GTX 102 is to turn on the paternal gene and produce UBE3A protein. The phase 1 clinical trial is a first-in-human study. It involves 4 monthly doses. It's a dose escalation study. There is no placebo control, and GTX 102 is given intrathecally by lumbar puncture. There's a screening period of up to 4 weeks, treatment of 4 monthly doses, followed by a 6-week follow-up period. The study objectives: the primary objective is to determine the safety of GTX 102. The secondary objective is to determine the pharmacokinetics. Pharmacokinetics is a measure of the drug in the blood and the cerebral spinal fluid. And then we're looking at a whole host of exploratory markers, looking at multiple functional domains of Angelman syndrome, looking at communication, sleep, behavior, gross motor, fine motor, seizures. The key inclusion criteria is a full maternal UBE3A gene deletion, age of 4 to 17, stable seizure control, normal kidney and liver tests, and able to tolerate anesthesia. The key exclusion criteria are any change in meds or diet to treat AS symptoms in the prior 3 months, inability to ambulate independently or with an assistive device or caregiver handhold. Any bleeding or platelet disorder would require intubation. The exploratory endpoints measure multiple domains. The question is, which test measures clinical change best? And actually, it's going to be a combination of these that are going to be required to understand the impact of GTX 102 on various functional domains. Looking at sleep through a variety of measures, seizures, communication, behavior, cognition, and motor. The study design, the dose escalation starts cohort 1 at 3.3 milligrams. The patients are treated, and after their second dose, a waiting period of 2 weeks, all that safety data is reviewed by an independent external data safety monitoring board. This is composed of 3 individuals: an expert pediatric neurologist with expertise in Angelman syndrome, a pediatric anesthesiologist, and a statistician. They review the safety data. They cleared those patients to continue treatment, and also for the next cohort, cohort 2, to start treatment. And this process was continued. In total, 5 patients have been treated to date. All patients were treated at Rush as it was open prior to the COVID pandemic. There's a 10-fold difference from the lowest to the highest doses tested. You can see here in cohort 1, 3.3 milligrams was the first dose, and they got as high as 36 milligrams. There was a 10-year-old boy and a 5-year-old girl treated in cohort 1. there was a 12-year-old boy and a 15-year-old girl treated in cohort 2 and a 5-year-old boy treated in cohort 3. So 3.3 milligrams was the first dose, 10 milligrams was the second dose. 2 weeks after that, the Data Safety Monitoring Board met, cleared enrollment in cohort 2. Cohort 1 patients continued with their treatment. Cohort 2 then continued after their second dose of 20 milligrams. 2 weeks later, the Data Safety Monitoring Board reviewed the clinical data and then allowed enrollment in cohort 3. The cumulative doses here are in the last row. They range from 20 milligrams to 105.3 milligrams. This graph depicts a plasma pharmacokinetics. It's a linear scale. The levels of GTX-102 in the blood were dose proportional. As expected, GTX-102 was not detectable 28 days later prior to the next dose in either the blood or the CSF. These graphs here are based off the patient's first dose. At this time, I would now like to turn it over to Dr. Barry Kravis to go over the clinical data. Thank you.